amazing CV I didn't realize till today. So he's the director, computational oncology and bioinformatics at Sage Bionetworks, and just in trained as an electrical engineer. Before that, I really you had a history and pre-med, and then Bachelor of Science in Electrical Engineering and PhD in Computational Biology at Duke. And also he started a company. He was the co-founder for Five Site Technologies in 1999 to 2005. So he has amazing resume and experience. And there's some sort of personal thing I want to tell you. One of the hardest thing I done in Raidu is running. But one thing we did together to run a 69 floors race together. So that was a quite experience. We did within 14 minutes.
Several times up and down, or very
Just down, all the way out.
One be a process.
Yeah. Anyway, so
And needless to say,
no. So anyway,
he's carrying less weight.
Thank you so much for coming, Justin, and I'm looking forward to hear you.
All right. Well, thank you very much, Indika. Thank you for the nice introduction and the invitation, and thank you all for attending this talk. The name of this talk is actually changed. If you can see, it was previously called the Consensus Subtypes of Colorectal Cancer. I promise I'll be talking about that, but I decided to go ahead and generalize the talk to a broader crowd, and to also introduce Sage Bionetworks. Many of you may have not have heard of us, although those who've been part of the DREAM competition should hopefully know us well. I know there's some in the audience who are kind of stellar participants, so we're very glad that you're here and that you participate, and I'm hoping many others will participate in some of the future challenges we have to offer. So as Indika said, I had the computational oncology group at Sage. We have other disciplines that we cover as well outside of oncology. I'll be talking about the oncology work we do. So we're located in Seattle, beautiful Seattle here, and we're located in the Fred Hutchinson Cancer Research Building. Yes, beautiful day. One day. The global warming is improving the weather in Seattle. You might think of returning. And we've been there since 2009. We were founded 2009 by this gentleman here, Stephen Friend. And the mission of this organization was to provide tools and environments to help improve reproducibility and collaboration in biomedical research. And not only do we build the tools, but we eat our own dog food. We actually do the research ourselves. And the goal is to really kind of shake up as Stephen would call it, the biomedical industrial complex, and think about different forms in which collaborations and incentives and patient engagement can occur. And so a lot of our efforts are with that motivation, although you'll see that some of our work is also quite traditional in the research space as well. As I said, we have tools. This is one of our tools called Synapse. It's a data platform for collaboration and data sharing. This is just an example from our colorectal consortium and the provenance graph of how this data was generated. I'm not here to sell Synapse, just to let you know about it. And here are the number of consortium and projects we're involved in, spanning progenitor stem cells, DREAM challenges, influenza. We're part of the TCGA pan-cancer effort as well as the ICGC, a large number of neurodegenerative projects, and then, of course, many oncology efforts as well. So I'm going to, before I go into the research focus of this talk, I want to just share a couple of interesting side projects that I've been part of that are kind of interesting and a bit orthodox. So this is work we published recently. It was, so the title here, Social Interactions for Enabling Research Communities. And the idea of this, so Frank McCormick, he was the AACR president 2 years ago, big researcher in RAS. He and Stephen were talking and said, you know, let's take advantage of the fact that you're president and do something a little different at this ACR. And they said, well, we have all these abstracts that people have submitted. Why don't we do some kind of interesting text mining exercise where we can try to find interesting relationships? So ACR gave us all these abstracts. I did some fairly rudimentary text mining to build this social interactome. And let me add that this was only done for people who are doing research in RAS. We didn't do it ACR-wide, that's too big, or with that 6,000 abstracts. We just concentrated on people doing research in RAS. And then we sent out a number of survey questions to these same set of researchers to understand, like, what are the major questions in their field? What are the unresolved problems? Where's the controversy? Where's there too much research? Where's there too little? And so between this interactum and this survey, we led a special session at ACR 2013, 2014. And it worked out quite well. ACR liked it so much, and there was such engagement in the group. In fact, we had a big ballroom, it was standing room only. They said we want to do this every year now. And so now we're going to do this at this upcoming ACR conference. This time it's going to be on MIC. So if there's any MIC researchers here, I hope you can attend. We're going to move on from RAS. So this is a kind of a fun project. And I'll add a couple words about the Dream Challenges. I know most of you know about this. If you don't, so the Dream Challenges are now merged with Sage Bionetworks. So that happened a couple years ago. So we're essentially one and the same. And the goal is to create an open crowdsourced competition to engage the community, engage people outside the biomedical community, engage electrical engineers, computer scientists, and to look at hard problems in biomedical research that can be quantitatively scored. I mean, it is a competition where we have to find a winner, so we have to have the right kind of data and the right set of questions. And so this is recently what we've concluded. We've had, this is the last round that we've had, competition in Alzheimer's, the gene essentiality prediction, that's trying to predict synthetic lethal using genomics. We've had the somatic mutation calling challenge. This is variant calling. This was led by Paul Boutros at OSER and Josh Stewart at UC Santa Cruz, and also one in AML. I want to give a quick, sure. It's very challenge dependent. So, for example, in the somatic mutation calling, it's very challenging. What is the true gold standard? So they had a simulation data set. So they have the Paul Boutros lab. He's able to generate synthetic genomes and add specific mutations to it. And so that's one gold standard. But of course, simulations are never very satisfactory. So they did obtain 5 real samples and had very high depth sequencing to generate a set of what they consider real candidates. And if, for example, in AML, this would be where you withhold a set of patients whose outcome is known, release some data where people can train, and then they test on this unseen data. So very kind of standard machine learning scenario. But what we do is we take the validation out of the hands of you all. Not that that you can't be trusted, but there's always this question: how do you objectively score people's predictions, and how do you do so in a way where you don't fool yourself? Because it's very easy. I'm a machine learner myself. I very often convince myself that I have the best algorithm, the best model, and lo and behold, I've overfit. So it's very easy to fool yourself. And so we set up these in a way that tries to overcome those limitations. Looking forward, here's the next round of challenges we'll have in the next few months. So continuing on the somatic mutation calling challenge, there's one now on tumor heterogeneity and evolution, one on RNA, and this is going to be looking at fusion detection. And then this is one that I'm going to be leading. This is the prostate cancer dream challenge. So I'll say a little more on that. So this is structured to predict 2 things: outcome in metastatic castrate-resistant prostate cancer patients treated with first-line docetaxel. So to predict overall survival and to predict treatment discontinuation due to adverse events, most likely docetaxel treatment. And the data comes from 4 pharmaceuticals who've opened up their data sets to us, where they're giving us the comparative arms of their trials. So 4 trials. In total, we'll have 1,500 patients in the training set, about 500 patients in the validation set. And we have, as lined up as incentives, we have a partnership with Nature Biotech, who will review the paper, the final manuscript. We have prize money. And so here's an interesting, this is new for this year. So who is familiar with AJCC, if you do cancer research? Okay, so there's no clinicians in this room. Okay, if you're listening, you might, you'd probably know AJCC. It's the American Joint Committee on Cancer, and they define the TNM staging. So you go in and you're staged as stage 1 or stage 2. So you know the criteria for your staging, and really what it measures is the extent of disease burden, is what AJCC really defines. And this year, for their 8th edition, they want to include, they want to expand their domain to looking at prognostic models more broadly. They want to have a suite of calculators in a sense. And these go directly into the hands of clinicians. So one of the things that we've always kind of wanted with the Dream Challenge is to have an application where the model can move directly into a translatable environment, into the clinic, where it actually has utility. So here we think we found an opportunity where AJCC has pledged to review any top winners that come out of this, and then get this translated into a calculator that could be deployed for all physicians to use when they're staging their patients. So very exciting, and this would obviously just be for patients in prostate cancer. And just real quickly, some other areas that we're active in oncology: neurofibromatosis, type 1 and 2, TCGA pan-cancer efforts. We're preparing another Dream Challenge to predict drug synergy, and this with AstraZeneca. We're working with AML labs where there's ex vivo screening and omics to predict patient response. We're working more on image analysis and getting more interested in mobile and sensor data. So that's kind of the kind of quick overview. So now I want to talk about, yeah.
Wouldn't they need more validation before they actually start applying it in the clinic?
Sure. Yes. Yeah. I kind of obscured that. So there's a whole process. In fact, I'm part of what's called the Precision Medicine Core. That's an AJCC committee that is basically all kind of a lot of the biostatisticians who get in a room and say, what's the level of evidence required? And we also interact with physicians, the surgeons themselves, pathologists, et cetera, who finally come to consensus of what a real prognostic model should be. And they're a very conservative group. So the level of evidence is actually quite high for them to actually endorse something. So this is kind of the first stage. There is a set of data sets that are owned by cooperative groups and others that we could also use for further validation. But it's at least the first step that if we'll recognize this model, now we'll apply it to the cooperative groups data, trial data, and see if it validates there as well. And again, yeah, and there's 2 trials. There's the Enthuse trial. It's over 2,000 patients. So we couldn't get access to it for the competition, but we'll have access to it in private to further validate these models. So yeah, I don't mean to suggest that it's a kind of a rubber stamp into AJCC. And at the same time, this is in kind of parallel to the TNM staging. That's really kind of sacrosanct, and that's not going to change so much. But now they're going to have a kind of in parallel these separate calculators. And because it's the first time, I think it's going to be it's going to be beta. I mean, clinicians will look at this, they'll evaluate and assess how good these are, but whether they get used in the clinic in a real way remains to be seen. Okay, so here's I'm going to kind of go briefly through this project here. I include it now because Indica was a part of this. We had many discussions about aging and what it means to have biological aging versus chronological aging. But this work was completed a couple of years ago. So we wanted to look at this phenomenon of chronological versus biological aging because we believe it exists. So here we have Sean Connery, who we think is a graceful ager. And then you have others who, through the same course of time, seem to have accelerated aging. So there's, sure, of course. But it does beg the question, you have chronology, which doesn't change. That time is very constant. But biological aging may undergo shifts. So the question is there such thing as a differential aging rate? So we looked at this question. we obtained 488 individuals who with whole blood and put them on methylation arrays. And I should add that this was that this data came from an aborted study looking at something else. They wanted to look at macular degeneration, I think, but they completely confounded the way they did the profiling. So, like, well, we have all this methylation arrays. What are we going to do with it? So they gave it to us and said, let's study aging. And this was shortly after Illumina came out with their 450K beat array. So we had this data. We had it profiled in the 450K. We had a lot of demographic information, age, and some other covariates. And we also obtained some exome genotypes on these samples as well. And when you look at the distribution of all the methylome, you know, here it's kind of bimodal, so you're either kind of hypermethylated or hypomethylated. Here's the age distribution. And when you look at pure correlation with just marker by marker with age, you find there's a lot of CpGs that are both positively and negatively correlated with one's age. So given that we saw that there was signal here, we thought, well, let's build a model that can predict one's age based on just the methylation profiles. So this is not the computer we used, but we did as a dependent variable is their actual age, and the independent variables were all the CpGs, 450K. And we, of course, did filtering and other things as well. And used an elastic net penalized regression model to fit the whole thing. And this is what we observed. We actually observed that you can predict age quite precisely just using the methylome. So here's our first cohort that we trained in, and what you're seeing here is the cross-validation results. It's not validating on the same samples that we're training on. And we observed that we observed a really strong correlation, an RMSE of 3.82 years. We did obtain a secondary cohort of 180 patients. Patients is the wrong word. It's sample, and see that we can recapitulate this model quite well. So we thought we had what was in effect a model of biological aging, with the assumption that on average people are going to follow their chronological age. It's the deviations from this fit where you see either accelerated aging, you know, above the line or slower aging below the line. At least. Well, we didn't have a prediction. We had no. Sure. Yeah, it's amazing how accurately it does follow, but it becomes circular if you try to second guess, you know, how far it should be, because we don't really know, because the concept of biological aging itself is still defined. But to try to answer that question, right? So we did look at well, first of all, the model was based on 67 markers. Most of the markers we found were disease-related or involved in processes that are associated with aging, DNA damage, Alzheimer's, oxidative stress. Here's one interesting gene that had a very high coefficient in our model, and also KLF14, which is the massive regulator known to be involved in obesity and metabolic traits. So we did have some patient demographics. So we asked, you know, does this differential, this accelerated or decelerated aging, is that correlated with any other covariate? So where we found the greatest signals, in fact, that males and females had differential aging rates. Males showing a higher acceleration than females, and you know, I just conjecture that maybe this explains why men don't have quite the lifespan women have. We did look at other areas as well: diabetes, smoking, BMI, and we were hoping that one of these might be associated with accelerated aging, but didn't observe it. We also looked at methyl QTLs and those that perhaps could explain accelerated aging as well. We found some interesting candidates that we validated. I'm not going to go into that. And so following our publication, soon after, just a few months later, Steve Horvath at UCLA came out with this paper here. It's a very extensive, in fact, you used the same model we had used, the elastic net, trained it on methylation and predicted on chronology. So very coincidentally, very similar study, but he looked more broadly at other tissue types and found that this model can be recapitulated in other tissues as well. And then he was able to actually do the study that we wanted to show that the disease phenotype correlated with accelerated aging. And he was able to observe this in the liver and found that obesity was associated with this higher acceleration of age. So it's nonlinear at younger than 20 years of age. So it's very linear from 20 upward, but there's a nonlinear transformation one has to do when you're looking below 20. Yes, I did. We observed that as well. I don't know enough to comment, but I do believe that there is kind of this accelerated aging process that occurs when you're younger and then levels out as you move beyond puberty into late adolescence. So now you can go to Wikipedia, and there's even a whole concept of the epigenetic clock, DNA methylation age, and so it's so the concept of biological aging, of course, because it's in Wikipedia, it must mean it's true. That there is this concept of biological aging. And we haven't done any more work on this since, but I think it's very interesting. And I know Indica has also done a lot of work in biological systems and age in the small in the sense. We don't have that kind of data. We did look at cancer. So we took our model, applied it to TCGA, first to the normal, so we can look at the different tissues, and found that in general we found a fairly good concordance of the age in TCGA and what we predict, but with a bit of a shift. And then when you go look in cancer, you tend to things get very, very complicated very quickly. Like in colon cancer, because of microsatellite instability and very strong changes to the methylome, it becomes harder to use that as evidence of accelerated aging because there's so many other variables that are now changing. But so the cancer might be the wrong place to immediately adapt that. And yeah, and those data sets, when they come available, I think it's very interesting to look at. But there's not a whole lot, unfortunately, to look at the moment. This is the only study I've seen that's actually looked at that question. Okay, so this work was done with Troy Idacker's group in UC San Diego, and Illumina was also a partner in this. Okay, so now I want to move into what I promised before, which was colon cancer. And it's of course a cliche to say that cancer is heterogeneous. We all know that. But there's different areas of heterogeneity that we can explore. And so I'm going to talk about these three areas here. The global molecular patterns, that's subtyping. There's the pathway heterogeneity, and then there's also prognostication outcome heterogeneity. And I'm going to address these in the kind of three vignettes. So the Colorectal Cancer Consortium is one where we look at the global molecular heterogeneity, RAS modeling work that we did a couple years ago, and then some future work that we're doing with AJCC. Okay, so let's move on to the Colorectal Cancer Consortium. As I said, cancer in general, and certainly colorectal, is a heterogeneous disease. Over the last couple of years, many independent groups have reported on molecular subtyping. And each group has kind of ascribed their own biology or interpretation to their subtypes. But what's not clear is there a concordance among all these subtypes? Is there actually agreement in terms of what people are ascribing to the biology? And so here's some of the papers that have been published in the last couple of years. And when you try to map all these different subtypes, this is what it looks like. So you have TCG at top, and here you have 6 groups. And some say there's 3 subtypes. Other groups say there's 5 subtypes or 6 subtypes. And we know that subtyping, there's no gold standard. How do you say how many subtypes there really are? It depends on how you threshold things. How many, what's your evidence for truncating your clustering tree? So we know that there's expected heterogeneity. But is there a way forward beyond this mess? And is there a way for us to make a very strong statement about what are the definitive colorectal subtypes? So what we did we formed a consortium among all the groups that you just saw. And they were committed to share all respective data sets, whether they have them in-house or they've curated the public data sets. And we were going to compare and validate all of the existing classification platforms. We're going to then conduct an integrative analysis across the pooled data sets and then define the clinical molecular hallmarks of these subtypes. And most importantly, we wanted this to be an example for all future subtyping efforts. Because there's many people who are doing this, many people publishing, but subtyping doesn't really find its way beyond the research manuscript. It doesn't find its way into the clinic, and I think a lot of that is because there's no good mechanism for defining a robust consensus that actually is translatable. So this is the first time that we've kind of seen this effort come together like this. So here's more or less the diagram describing how this operated. Here are the 6 groups that participated. SAGE was the kind of, we were Switzerland. We were the independent group that can, because recall these are competitors. They've all published and they're all trying to play against each other, but they needed a neutral party like SAGE, who doesn't have our own classifier. We brought all the data together, normalized it in one consistent way so that we weren't looking at differences there. And then all the groups applied their algorithm to all the pooled samples. We developed a method to identify the consensus subtyping, if there was one in the data sets. From that, we built a classifier and then did all the characterization, the downstream interpretation of the subtypes. So here's the data sets. We were able to pool over 4,500 samples across fresh frozen and FFPE, Affymetrix, RNA sequencing, actually a lot of public and proprietary data sets. And only one, yeah, that's just the TCGA data for that. We didn't use splice isoforms. We are studying, that's ongoing work where we're looking at the isoform differences across these subtypes. But that's not, that's still very early work. This is distributed across all stages, but primarily stage 2 and 3, as one might expect. And same distributions with respect to microsatellite instability samples. And we also had follow-up data for many of these samples as well. So this is kind of how you thought about how you define a consensus subtype. It's basically just looking for common patterns across all the 6 groups. So if you see a common pattern here, we might call that consensus molecular subtype 1. And another pattern, we call it 2. And if we didn't see any pattern, we call it unclassified. And to identify these clusters, we use just a random walk on a graph where the graph is created across all these subtypes across all the samples. I won't get into the details there, but when we run this algorithm through many bootstraps and trying to be robust as we can, this is what we observe. And so each of these bubbles represents a subtype from a particular group. And what this told us is that there were 4 robust subtypes that emerged when you looked across all these groups. And here's another way of observing that here. You can see here's CMS1, and you see kind of a common color across the groups that define these different subtypes. And they all have different number of their own subtypes across the groups. But you can start to see how there's a common pattern across here. And of course, there were a number of samples that didn't show any concordance at all. And we just called these to be unclassified. And this was about 20% of the samples that remained unclassified. Here's another way of looking at this in kind of network space where we can, each dot represents a patient. And we can cluster this in a way that, again, allows you to see, we simply color these clusters according to our subtyping, our consensus subtyping. And many of the unclassified fall in between these subtypes, as you might expect. So this is just another way to visualize that. You simply load the data in Cytoscape and let it cluster. Yeah. Okay, so now that we have this consensus subtype, and we also built a classifier where we took those 20% that were unclassified. Our classifier was able to reclassify half of those. So now at the end of the day, we only have 10% of the samples that ultimately are unclassified. So now it comes to interpretability. So we have these subtypes. What do they mean? So we had all this demographic and clinical information, other molecular information. So we just started doing very basic statistics, differential statistics. We find CMS1 is more enriched for older patients and is also enriched for female females. And CMS4 is slightly enriched for stage 4. In CMS1, we see it's enriched for the right side of the colon, and also CMS1 is enriched for the microsatellite instability. In CMS1, again, we also see BRAF enrichment, and we know that BRAF mutation and MSI status are awfully are linked. And here we see that KRAS mutation is enriched in CMS3. We did a lot of correlative analysis with all the different kind of omic data available, especially with TCGA. We can look at copy number instability, chromosome instability, SIMP status, many mutations that we did correlative analysis, copy number, methylation, microRNA, and then the recent proteome data that was published in Nature about a year ago. We also did correlative analysis with that as well. I'm not going to get into those specific results because I think the main interpretability of the subtypes comes here when you do pathway analysis. So here we see in CMS2. CMS2 is kind of the canonical. This is the Bert Goldstein model of colon cancer, Wnt activation, MYC activation. CMS4 is the mesenchymal phenotype, where we see TGF-beta activation, matrix remodeling, stromal infiltration. CMS1, we also see JAK-STAT, proteasome activation. CMS4, we see VEGF integrin. CMS1, we see immune infiltration. And so CMS3 was always kind of this question mark. What is CMS3? We know it's enriched for KRAS mutation. And so here this is where it really became obvious what CMS3 and with high metabolic activity. So this really allowed us to characterize this in a way that it hadn't been before. We did overall survival analysis. CMS4, this is mesenchymal, has a much worse outcome than the other 3 subtypes. And this was true even with multivariable analysis accounting for mutations in MSI status and age and other demographic information. CMS4 is also much worse outcome for disease-free survival. And so this was very interesting when we looked at survival after relapse. So after you have a primary that comes back, and we see that here CMS4 has a much worse outcome than CMS2, and then CMS3 and CMS4, this is mesenchymal and the metabolic, are kind of in the middle there. So again, something very unexpected that came out of this work. Yes, this is the CMS1. So this is the immune infiltration MSI high class that does much worse. So yes, I mean that's a good point. So that's actually we don't believe the primary changes much, but the question is when you look at the metastatic site, does that change? And that's actually what we're looking at now. That's ongoing work. So here we are with what we call the taxonomy of colorectal cancer. CMS1 is the MSI immune, CMS2 is canonical, CMS3 metabolic, and the CMS4 mesenchymal. And so this is the taxonomy that the expert group has agreed on, and what we hope will be the future basis for patient stratification in clinical trials. In fact, we're already working with a few companies, pharmaceuticals, who are deploying this in that context. Yeah. So yeah, so APC is kind of believed to be an earlier event, and so it's found in over 70–80% of most samples. And we looked and we didn't really define any kind of strong association there. Same with PI3 kinase. That tends to co-occur with KRAS. So one might argue that CMS3 has a slight enrichment for PI3 kinase. Was there another gene that you mentioned? P53. Oh, yeah, that also was frequently mutated, that we didn't see any strong association. So that's kind of represents a body of work that's complete and is now under review. Ongoing work is really how do we make this useful? These subtypes, if we want to make them useful, we have to find how they associate with treatment or patient response. Patient-derived xenografts is one area that there's a lot of activity on. So we wanted to look at whether the CNS subtypes were recapitulated in the PDX model. And if you just take, so this is a patient. So here we have in blue are the primary tumors, and here in red are the PDX derived from these primary tumors. And you see when you cluster them, this is gene expression data, they immediately separate. So there's obviously a very strong difference in the PDX and the primary tumor. But the question is we take our CMS genes that are used in our model. The question is, how does that affect it when you go from PDX, from tumor to PDX? And by and large, for the CMS 1, 2, and 3, we see that the important genes shown in red tend to correlate across these between the tumor and the PDX. The CMS4, which is the mesenchymal, the stromal infiltrated subtype, where we see that there's actually a loss of that stroma here in the PDX model. And there's going to be actually a really interesting paper coming out in Nature Genetics soon, which shows that the PDX is the mouse. So if you take the RNA-seq and then you now align it to the mouse genome, you can actually find a lot of the stromal genes rediscovered and could act as a substitute for the stroma and CMS1. Yeah, so if you go back here and you basically remove the major effects between PDX, if you like regress and take the residuals between the PDX, and you redo this clustering, the PDX and the primary pair up. Yeah, so that's still preserved and can be found here. I'm sorry, what was the question? So like different passages of the PDX? Oh, I see. Interesting. No, I don't think that's been observed, but I can't be. I'm not 100% sure, but I don't believe. So the person who did a lot, who's going to be publishing soon on the PDX, I think they commented that there was not an absence of. They found that all CNS types were represented there. Once you accounted for this loss of stroma in the CMS4, but they were found more or less in the same percentage. Okay. What time is this? 4:30. Okay. So I think a time to run through some of the RAS modeling we've done. This is at the pathway level now, looking at heterogeneity in colorectal cancer. And the idea here is that, so then we like to think that you have an aberration, you have a RAS mutation, you have the BRCA mutation, then that pathway is somehow activated in a very discrete way, in a bimodal way, whether it's either on or off. But we know that there's a lot of modulators of a pathway that can either attenuate or make the pathway even more active. So there's this concept of NIS, RASness, BRCA-ness, PI3 kinase, right? So it's this idea that it's not really discrete, but it is continuous. And if you look at, if you try to find surrogates for the pathway activity, then you'll be richer in information than if you just look at the aberrational level. So we believe this as well. So our idea was that in colon cancer, that the canonical mutation of KRAS 12, codon 12 and 13, is the most canonical way of thinking about RAS activation. But that there can be other aberrations that may enhance or mitigate the activity of this pathway. So we wanted to develop a quantitative readout of RASness. And people have done this before, but not in colorectal cancer. So there's RAS signatures developed mostly in lung. And so we took those and we asked how did they do in being able to differentiate KRAS mutants versus wild type samples. And they actually didn't do that well. So this led us to develop our own model, where here we trained, so similar to the methylation and aging concept, where we want to develop a predictive model that can predict KRAS 12 and 13 mutations using gene expression data. We had 320 samples to train our model on. And we ended up with a fairly robust model. So here we trained it, and here's the validation data sets we tested it on. So here's the area under the ROC curve. So pretty good accuracy. We then asked how well did this model do on other diseases that have frequent KRAS mutations, like lung endocarcinoma. And you see not nearly as good, showing that this is very colorectal specific. And same with endometrial. So now we wanted to ask, now that we have this quantitative of RAS activity, so how does this compare with when you look at the different variants of RAS mutations in colon cancer? So we were able to do this with TCGA. So here's our RAS index score. 0 is low RAS activity, 1 is high RAS activity. And so here we're able to plot all the samples in the colon cancer TCGA data set. Here are the variants in KRAS. Blue is BRAF, and here in yellow is NRAS. Well, not necessarily. There's a suggestion that 13D mutations are not as potent, so that perhaps they might even be eligible for cetuximab treatment. So this now this is I don't think strong evidence that there's less RAS activity in G13D, but we see that there is an interesting distribution. Certainly, BRAF tends to have a lower RAS activation. NRAS has very comparable to KRAS, and we see some very rare mutations here. These are only N of 1, but it may indicate that these are not actually activators of the oncogene. And so this might be able to, one way of differentiating between true activating mutations and passenger mutations. Now notice here, here are the wild type, the so-called wild type. But there's a number of them that have very high RAS activity, despite no KRAS, BRAF, or NRAS mutations. So we did look in this group and look for other mutations that might explain it. We weren't really powered. We have some very interesting hypotheses, but we weren't really powered to detect any obvious candidates that would suggest activation of this pathway. We next asked whether this RAS score can predict drug sensitivity. So we applied our model to cell line data. So these are two cell line panels, the CCLE and Sanger. And we found that the drugs that are most correlated with our RAS index score were the MEK inhibitors. I guess not surprising. Although we interestingly found that the MEK inhibitors were not correlated with RAS mutation. So you wouldn't find it if you just looked at the mutations. You only observe it when you look at our predicted RAS model. So predicts sensitivity to MEK, and also it turns out gamma secretase. But we didn't follow that up. We did look at a NOX inhibitor. We did look in vitro whether exposure to MEK inhibitor would cause the reverse action, where you'd actually see RAS activity go down. And that's what you observe here. So here's 3 cell lines, all with either a BRAF or KRAS mutation. Here's the MSO, and here's the transcriptional output of our model, showing that RAS index is lowered when cells are exposed to this MEK inhibitor. These are both MEK inhibitors here, and this is an EGFR inhibitor, where you see no change. And then we went to the xenograph domain. One xenograph is KRAS mutant, and so the MEK inhibitor again shows that it can reduce the RAS pathway activity, shown by our model here. And as we would expect in the KRAS wild type domain, we see no effect of the MEK inhibitor. In fact, it actually has a small perturbation to the pathway, as if MEK inhibitors somehow cause a slight increase of RAS activation, which actually has been reported in the literature. Okay, finally, we looked at whether our model can predict response to cetuximab, so anti-EGFR therapy. And so we know that, so cetuximab is only given to patients who are KRAS wild type. We know it's not effective in KRAS mutant patients. So we just looked in the wild type patients and asked, in that domain, can we predict patients who are more likely to respond, have better outcomes? And we found that in both 2 data sets, one a completely blinded validation set, that in both cases, the high risk activity was indicative of a worse response to cetuximab, as measured by overall survival. So that summarizes the RAS work. I want to make sure we have time for discussion. It's 4:22. Sure. Yeah. So this is a different domain. This is first of all, this is metastatic colon cancer treated with anti-EGFR therapy. So So before this is, we were in the CMS group, we were simply prognostic in terms of outcome. This is based on actually response to therapy. So it's a kind of a different domain. Now, we didn't take our subtypes and apply them in this context. This is kind of older work, but we may expect to see that the CMS3 subtype has a less response because it's a higher reactivity. It's less likely to respond. Sure. Absolutely. Yeah. I mean, that's something that we haven't done. I would again expect that we'd see enrichment in the CMS3, which is the KRAS high CMS subtype. Um, yeah, we actually haven't applied this in that domain. And are you talking about the AJCC? So AJCC is not in the business of predictive models. So for those not familiar with cancer, there's this odd distinction between prognostic and predictive. Predictive means response to therapy. Prognostic means just overall survival based on whatever baseline features you might have. So AJCC is purely prognostic. So even though clinically, I think most of the interest is response to therapy, um, they wouldn't be interested in the RAS model itself. Yeah, so yeah, the AGCC is the College of Surgeons. So they're the ones who are getting in very early and cutting it out. So they're not the medical oncologists. Okay, so I'll just add one. I can summarize this fairly quickly, and that is, so AJCC, I talked about their previous interest in these precision medicine models. Separately, they've approached me in the space of colorectal cancer to ask, we'd like to see whether there's sufficient evidence to add biomarkers for the first time ever into AJCC. And specifically, they wanted to look at these three biomarkers, MSI, BRAF, and KRAS, and to ask whether those should be prognostic markers added into the TNM stage. And again, this is a very conservative group. They have a high level of evidence, and so to pull this off, we're going to have to get the widest accumulation of data together to really see if there's sufficient evidence for this. And so, here's what we've been doing. We've been going after many groups in their data set. So a number of clinical trials and prospective cohorts. And now we're at the point where we have about 10,000 samples accumulated where we also have MSI, BRAF, and KRAS annotated. And it's a pretty heterogeneous group. Some of these are family registries, prospective cohorts, clinical trials, and a different stage representation. So anyway, this has been a very, to say the least, it's been a very challenging endeavor trying to extract real clinical data with outcome metrics. But we think we're at the number now where we can now begin the modeling and see if there's evidence. And so we have, we don't have the full 10,000 yet, but we have about 3,000 samples, and we've done some preliminary modeling. So we're starting with the TNM, which is the baseline. So that's just the tumor, the number of lymph nodes, whether you're metastatic or not. And then asking, okay, so if you add MSI and BRAF and KRAS, how much does that improve your predicted performance? How much better can you do in prognosis? So here's what we've begun to observe. So here's the TNM. We're using concordance index as our metric. We find that when you add MSI and BRAF, that tends to improve, and finally KRAS does even better. Doesn't do so as well when you're measuring disease-free survival. So we can also add TNM, and we can also add other clinical covariates like age or site, or these, what you might expect that they'd want in these models. It doesn't have as much distinctive ability, but still we see a trend, at least with the concordance index, that MSI and BRAF, and then finally KRAS do improve the final model. We can also look at time-dependent AUC. Here again, the TNM. Here's the TNM with MSI BRAF, and finally when we add KRAS, we see a fairly strong trend in terms of improving prediction of overall survival, and in this case also disease-free survival. And I think we'll leave it at that. So we're still at a very early stage. The 8th edition comes out in 2016, so a lot of work in the next few months to get all the data together, harmonize it, and to begin to assess whether these models are going to provide the level of evidence that's required. So we can't do that because the subtyping system is based on gene expression, and gene expression. So for an AJCC metric or calculator, it's going to have to be on biomarkers and other covariates that are routinely collected, and so that kind of rules out the gene expression domain. Now we are working on a small platform with NanoString that might be more widely deployed, but still I don't think it's going to be. So AJCC, so you have to be able to apply it in any context. Mm-hmm. Yeah. We can compare, and we do find that the CMS are predictive, independent of these markers here, MSI, KRAS, BRAF. So they do provide information over and beyond what these markers provide in terms of outcome. But it's just not practically deployable. That's right. Yeah, some of these are. That's right. The SIMP? Yeah, we about half the data has SIMP status as well, and I think there is evidence, but I don't think we have enough samples in the way we have with MSI, BRAF, and KRAS. Okay, so let me just acknowledge some key members of this work. So Rodrigo Diepstein, he's a medical oncologist at Vall d'Hebron, who's been the last 2 years at Sage, and never wants to go back to be a clinician. He wants to become a bioinformaticist. He's been instrumental in a lot of this work in colorectal. Most of our partners, especially in the colorectal domain, have been European groups. So we have French, Netherlands, UK, Vall d'Hebron, Belgium. Our collaborators here in the States, so the Mayo Clinic, and OHSU with Joe Gray, MD Anderson, and Renee Bernard's University of Michigan. I will finally add that Sage is hiring. So any postdocs or graduate students who are near completion, we're very interested in talking to you. So thank you very much. Yeah.
About those 6 groups who previously had different classification systems, you invite them together to, I assume they are still friends because you give them a task, which is to forget about their own data set, work on this joint data set. And you ask them to class assignment based on their old system. You didn't ask them to do ab initio discovery.
That's right.
So it's not so much of a challenge for them to redo classification, redo class discovery per se.
Yeah.
Right?
Yeah, it was a very simple task in a sense. Once they get beyond the problem of agreeing to share their data and agreeing that whatever we find is what they're going to finally support.
Right. And given how large this overall group is, I assume the conclusion will have its gravitas, will have a longer lifetime. And then you want to have the committee to further evaluate. It probably will publish well. Do you have plans to establish a smaller set, such as PAM50, so that it's really easy to use?
Yeah, so that's what we're currently working on. We're talking to a few diagnostic companies. Agendi is very interested in licensing this, although we're not thrilled with their platform, so we're talking to NanoString as well. But even at the end of the day, even if you can make a platform, it's still always the question, okay, what's the clinical utility? I think this is important in the research domain, and the clinical trials companies that are running clinical trials are also very interested in being able to salvage their drugs and finding that they respond in a particular subset. But just for your average clinician, I don't think they'd be very interested in this per se.
For BRAF mutations. We have something for BRAF patients, and we know that you have to do something about KRAS activation, either not treat those patients with the EGFR receptor inhibitors, or find a way to knock down KRAS activity. And otherwise, it's prognostic, it's useful for counseling and discussion, and it's certainly a base for further discovery. But I would say you have 4 categories plus a 5th, the way you presented it, really unclassified. And one teaching point, Justin said multiple times how heterogeneous cancers are. And I urge you all to use the term cancers in the plural and the verb are instead of is, so it's clear that you understand the heterogeneity of cancers.
Thank you. Nice talk. Could you please explain a little bit about predicting the drug synergy? You said one of your projects is prediction of drug synergy.
Yeah, so we're currently working on a pilot. So as you might imagine, pharmaceuticals are very interested in understanding their own drugs. And so they run these drug panels, large number of cell lines. They do the monotherapy, look at the I350, etc. And now they're starting to do all the combination screens. So AstraZeneca is sitting upon this big data set where they have many tens of thousands of combination screens that they've looked at. And they don't know what to do with it because they don't really understand it. And I'm not sure anyone really understands it. So we're partnering with them to host a challenge to see whether what are the thought process that we have to go through to think about the synergistic behavior with drugs. I don't think it's going to be very straightforward. It's not going to be just basically looking at the omics in a naive way. It's going to be thinking about network structures and pathways and how drug synergy is likely to occur. It sometimes could be completely orthogonal pathways, or completely because they're sequential and linear, and you have to target kind of upstream and downstream. So it's a hard problem, and we're going through the exercise of just evaluating whether we even have a robust enough data set to host the challenge. So that's the hope.
People wanting to make a comment? Go ahead.
Maybe what the data sets are going to be. Is copy number data going to be involved in that at all?
It's not going to be copy number. I think it's strictly looking at DNA variants. And so this is, unfortunately, I don't know a lot. This is going to be led by Paul Boutros and Josh Stewart. I haven't really been sitting on their calls, but it is to look at, to be able to look at clonal evolution, for example, and understand. And some of this is in silico, by the way. So in fact, I think it's all in silico. They're going to be able to make permutations, perturbations that would follow some kind of evolutionary trajectory and see if people can recapitulate that. But I unfortunately don't know the details.
So I want to pick up on your very good question about synergies. I think it could be dissected for starters like this. There are at least 2 different reasons, strategies for synergies. One is to get higher efficacy at the point of initial treatment. So understanding the pathways involved and the ones you can target simultaneously should be effective, and that's credible and rational. It's not just statistical random testing, if you look at it that way. The second reason to worry about synergies is to survive mutations that cause resistance. And this is a predictable effect, just like with microbes. It is shocking that we've had this strategy in clinical oncology to treat with one drug when we know that cancer cells are very likely to mutate and become resistant. So to survive resistance, it's very important to target at least 2 different sites, 2 different networks or pathways to maintain control of the cancer from growing. And that really should be separated from the first goal of efficacy.
Yeah, and unfortunately, I don't think we're going to be able to look at that second question, even though I think it's the most clinically relevant question today with targeted therapy, because we don't know how we don't know all the mechanisms by which the tumor will evolve to escape sensitivity to a single drug. And we don't have the data to evaluate that at this point. And then there's a third, which is how do you decrease the toxicity the idea is that you can give two drugs at lower doses and have higher efficacy than a high dose in a single drug, which can be highly toxic and have a lot of
The work of Xiaoman Wang, which is instead of trying to knock down hard a particular target and another target, to try to make a drug that works on the protein-protein interaction, like P53 MDM2. And at a much lower dose, you can interfere with protein-protein interaction than knocking out the activity of either. This has been clinically useful already, and it's a general strategy.
First, to increase some sort of a heterogeneity, to increase some the drug to increase entropy. There's idea that if you have more, so what I'm saying is, can you? I don't know much about cancer, but can you drug that accelerate and then attack, like a hybrid way of attacking the problem? Just not.
I'm not sure I understand. Because what's happening with the drug is you're selecting, usually what the problem with resistance is that you're selecting particular.
Yeah, I'll tell you later. So it's not good.
Okay, thank you very much.